Let's start today's session. Before start the new topic, let us recall about the topic that we have covered in previous lecture. So, in previous lecture, we have discussed about mainly the pressure which involved with the gaseous exchange, and also we have discussed about oxygen-hemoglobin dissociation curve and how the oxygen-hemoglobin dissociation curves are influenced by different factors. And we also discussed about the P50 of oxygen-hemoglobin dissociation curve. Let us brief again about the dissociation curve, and after that I will start the new topic, that is about the carbon dioxide transport. So, as I mentioned, that oxygen-hemoglobin dissociation curve actually explains about the relationship between the partial pressure of oxygen and partial saturation of hemoglobin. It explains about the affinity between hemoglobin and oxygen. This affinity between hemoglobin and oxygen are influenced by several factors. If you see the structures of the hemoglobin, you will find two different states where oxygen binds with hemoglobin. One is called T state, which is called tan state, and tan state is the state in which the affinity between oxygen and hemoglobin is very less. And another one is R state. The R state or relaxed state, it is the state in which the affinity between oxygen and hemoglobin is more, right? So here both the T state and R state are influenced by different factors. So the factors, as you already know, the concentration of carbon dioxide or partial pressure of carbon dioxide, then H plus ion concentrations of pH, temperature, and some metabolite called 2,3-BPG or organophosphate. Along with this, the other types of hemoglobins also affect the oxygen-hemoglobin dissociation curve, like myoglobins or natures of myoglobins also shows the different level of affinity with oxygen. So if we see the effects of all those factors, how can we assess the effects? To assess the effects or to assess the affinity between oxygen and hemoglobin, we need to know about the P50 value. So what is the P50 value? P50 value, it is the value in which the 50% of the hemoglobins are saturated with oxygen, right? So if you see the P50 value, it is around 26.6 millimeter mercury, but in certain conditions this P50 value changes. How can we measure the changes of P50 value? By seeing the graph. In some cases, the graph shifted towards the right side of the normal curve, and in other cases it is shifted towards the left side, right? For example, this was partial pressure of oxygen and this was partial saturation of hemoglobin. So the normal curve was sigmoidal in pattern. In this curve, we have discussed about the P50 value. If it is 100% saturation, and these points will be the 50%, right? If you see the x-axis, so it was around 26.6. Now, as I have mentioned, what are the factors? The factors are PCO2, temperature, H+ ion concentration, OPH, then certain metabolites or organophosphate, that is 2,3-bisphosphoglycerate. So in normal condition or resting state, it was this, means in this partial pressure of oxygen, 50% hemoglobins are saturated with oxygen. So now the conditions was increase of all those factors affects the hemoglobin structures. Actually, if you see the relationship between carbon dioxide with hemoglobin and oxygen with hemoglobin, you will find the difference. In presence of oxygen, hemoglobin shows more affinity towards oxygen in comparison to carbon dioxide. But in presence of more carbon dioxide in the media, the hemoglobin shows more affinity towards carbon dioxide, right? It means carbon dioxide or partial pressure of carbon dioxide, when it is more in a certain conditions, like in systemic capillary or concentration in pulmonary capillary, you will find sharp difference about the binding of hemoglobin with both the oxygen and carbon dioxide, right? So here, if the condition changes, the P50 also changes. If it is shifted towards the right side, it means the affinity of oxygen with hemoglobin is less. Means the hemoglobin will release or unload more and more amount of oxygen which is received by tissue, that is required for cellular respiration, right? If P50 shifted towards the left side, means if graph shifted towards the left side of the normal one, it means the affinity between oxygen and hemoglobin is more. The oxygen tightly binds to the hemoglobin molecule, right? So in these particular conditions, the loading of hemoglobin occurs so that cells will not get sufficient amount of oxygen for cellular respiration, right? So what we have seen with the increase of all those factors, this is just reverse of this H+ ion, increase of all those factors leads to the shifting of the sigmoidal curve towards the right side. Means there is a decrease in affinity between oxygen and hemoglobin, right? And if the affinity is less, so that unloading of oxygen should be more, cells will get more and more number of oxygen to perform cellular respiration to generate energy, right? At the same time, if all those factors or concentrations for all those factors are less, right, the graph will shift towards the left side. It means the affinity of oxygen with hemoglobin will be more. So the loading capacity of hemoglobin will be high, unloading capacity of hemoglobin will be less. The cells will get less amount of oxygen to perform cellular respiration, right? Now, if you compare the hemoglobin molecules with the myoglobin molecules. Both contains the globin protein, both has the capacity to carry the oxygen, right? But still, if you see the dissociation curve for both the myoglobin and hemoglobin, you will find sharp difference in P50 level of both the myoglobin as well as hemoglobin, right? So why? Because the affinity between the myoglobin and oxygen is much more than the affinity between hemoglobin and oxygen. So if the affinity is more in myoglobin, so that with very less amount of oxygen would be required for the fully saturation of myoglobin, right? In comparison to hemoglobin. It means for full saturation or 100% saturation of hemoglobin needs more number of oxygen molecule, or unloading of oxygen from hemoglobin will be more in comparison to the unloading of oxygen from myoglobin, as because the affinity between myoglobin and oxygen is much more than the affinity between hemoglobin and oxygen. And here myoglobin played an important role in certain cases. So what is the case? Case is in exercise state. During exercise we need more amount of energy, right? So here myoglobin acts as a storehouse, right? It can store the oxygen molecule which can be supplied in a certain condition, like during exercise, when there is a less supply of oxygen by aerobic respirations. So here myoglobin actually helps to slow down the anaerobic process, right? So these are some factors that affects the P50 value, right? These are some factors that affects the affinity between hemoglobin and oxygen. Now comes to the next point. As in previous class we have discussed about the various type of pressure that is essential for the gaseous transport. So here we will see the importance of those pressure during carbon dioxide transport, right? As you know, the partial pressure of carbon dioxide in arterial side and partial pressure of carbon dioxide in venous, there is a difference. Similarly, if you see the total carbon dioxide content in arterial end as well as in venous end, there is a difference in carbon dioxide content. Now question is, what is the pressures in arterial end and what is the total content of CO2 in arterial end and in the venous end, and how this partial pressures in both the end helps in the transportation of carbon dioxide from arterial end to venous end and ultimately from systemic capillary to pulmonary capillary and from pulmonary capillary to outside the body, right? Suppose this is arterial side, this is venous side, right? So what is the characteristics of arterial side here? The partial pressure of carbon dioxide in arterial side is 40 mmHg and total carbon dioxide content in this side is around 48 ml per deciliter of blood. See here in the Benson, the partial pressure of carbon dioxide is around 46 mmHg, and total carbon dioxide content is around 52 ml per deciliter of blood, right? Now here the transport of carbon dioxide are mainly carried out by hemoglobin molecule. Along with hemoglobin, if you see the transport process of carbon dioxide, you will find there are 3 different mode of transport. The first one is called the bicarbonate form, and second one is as carbamino form, as dissolved state. So there are 3 different mode by which carbon dioxide can be transported. And if you see the percentage of this transport system, the most of the percentage of carbon dioxide transport is carried out with the bicarbonate form, right? It is around 90%. In some cases you will find 75%. You will see in different books it is written in different way. But we can say the most of the percentage of carbon dioxide are carried out as a bicarbonate form, and carbamino form, that is around 5%, and in dissolved state it is around 5%. Now, if we discuss about the first one, before discussing about the transport of carbon dioxide as a bicarbonate, we must know about the functional characteristics of hemoglobin, because hemoglobin is the most important one here. Most of us know about the main functions of hemoglobin. Most of us know. That is the transport of oxygen as well as transport of carbon dioxide. But other than these two, hemoglobins plays some other roles. So what are the other functions that is carried out by hemoglobin? Yes, 10th standard students also know these two functions that is carried out by hemoglobin. Other than this, if you see the structures of hemoglobin, you will find there is lots of amino acid sequence in the globin chain. And this amino acid sequence carries some negative charge. If you see the charge of histidine, you will find the negatively charged histidine present in the globin chain. And this negatively charged molecule always attracts towards the positively charged molecules. Right? So here, the most important positively charged molecules that binds to the hemoglobin is H+ ion. Right? It means hemoglobin not only acts as a carrier of both the oxygen and carbon dioxide, it also involves with the binding of H+ ion. So why the binding is important? As because it helps to maintain the pH of the blood. It involves to maintain the acid-base balance, right? It means hemoglobin acts as a buffer system, right? This is the third function. Other than this function, hemoglobin also played another important role. So what is this? You have heard about the terms nitric oxide, right? During the inhalation, along with the oxygen, we inhale some amount of nitric oxide, right? This nitric oxide is also carried out by hemoglobin, right? So what is the functions of this nitric oxide? Nitric oxide acts as a vasodilator, right? It involves the vasodilatations of small arterioles. So these four are the most important function that is carried out by hemoglobin. Now, what is the exact mechanism of carbon dioxide transport? Suppose these are number of cells, tissues, right? Due to metabolic activities into those cells, they continuously produce more and more number of carbon dioxide, right? Now, suppose this is interstitial fluid, right? This is interstitial fluid and this is the cells which produce the carbon dioxide. If you see the concentrations of carbon dioxide in these two sides, or even if you see the concentration of carbon dioxide in plasma, right, or plasma fluid and tissue, the concentrations is more here. So obviously, according to the law of diffusion, the gases will diffuse, right? If you see the rate of diffusion, or if you compare the rate of diffusion between oxygen and carbon dioxide, if you compare the rate of diffusion through plasma membrane for both the oxygen and carbon dioxide can diffuse more rapidly because it is more soluble than oxygen. It is around 25 to 27 times more soluble with the lipid in comparison to carbon dioxide, right? So this carbon dioxide easily can diffuse from its higher concentration gradient to lower concentration gradient, right? And ultimately from interstitial fluid it will go into the plasma, right? So here in this RBC contains a special enzyme, and most of us know the rule of enzyme. Enzyme accelerates the biochemical reaction. Enzymes are very specific. Enzymes are protein in nature, right? Here, the most important type of enzyme present in the RBC is carbonic anhydrase, right? And this carbonic anhydrase enzyme accelerates the reactions is around 5,000 times faster than in absence of carbonic anhydrase enzymes. So here, during the transport of carbon dioxide, this carbon dioxide first react with water molecule, right, in presence of some enzyme that is called carbonic anhydrase, right? And it produces carbonic acid, right? This carbonic anhydrase enzymes not only involves with the production of carbonic acid, it also involves with the breakdown of carbonic acid, right? So here this carbonic acid is again breakdown into some ion, right? That is the proton ion and bicarbonate ion, right? Now, these reactions these reactions are due to increased rate of these reactions inside the cells, there is a generation of more and more number of H plus ion, right? More and more number of H plus ion, which gives the medium or which needs to the acidic state, right? So to maintain the balance or to maintain the pH of blood, it needs to either remove the H plus ion or it needs to bind with some other molecules so that the pH can be maintained. So here this H plus ion, suppose this is the hemoglobin chain. In this chain there are some histidine molecule having negative charge, right? So that this H plus ion will bind with this negatively charged molecule, right? Means this H plus ion will react with hemoglobin, then form HHB, right? So here, due to the binding of H plus ion with this hemoglobin, again there is a changes in the pH of the medium, right? Because now only H plus HCO3 is remaining here, right? So cells needs to blood needs to maintain the specific pH so that carbon dioxide transport will be proper. So here, to maintain the pH, if you see the cell membrane of RBC, in the cell membrane of the RBC, there is some exchanger called anion exchanger. So this anion exchanger here involved with the exchange of two different anion, that is chloride ion and bicarbonate ion, right? So here in this membrane there is an exchanger which involves the exchange of ion. So due to higher concentration of HCO3 minus here in the inside, through this exchanger it will come out, right? At the same time, the exchange basically occurs with the chloride ion, and this exchanger is known as, or exchange between bicarbonate and chloride is known as chloride sheet. So why this chloride sheet is important? It is important is because to maintain the pH only, right? To maintain the pH only. We have already seen in oxygen-hemoglobin dissociation curve, if there is a high H plus ion concentrations of low pH, it affects the affinity between hemoglobin and oxygen. We also have seen if there is a low pH and high H plus ion, it also affects the affinity between these two molecules, right? So to perform the proper functions of the cells, it needs to maintain the proper pH. So how to maintain the proper pH? So here chloride bicarbonate exchanger helps to maintain the pH of the cells. Now see this part is basically carried out into the systemic capillary. Now if you see as I have already mentioned, in pulmonary artery, what was the concentration of PCO2? What was the PCO2? That is around 40 mmHg. And what was the carbon dioxide quantum? It was around 48 ml per deciliter of blood, right? Now here in the veins, it was 46 mmHg, and carbon dioxide content was, that is 52 ml per deciliter. Now see, whenever blood passed through the capillaries, here in the arteries, the content was 48, but when it reached to the vein, now content is 52, right? There is an addition of 4 ml per deciliter of blood. It means the bicarbonate system carries 4 ml per deciliter of blood. Means the carbon dioxide that is diffused from the tissue to the plasma is 4 ml per deciliter of blood, right? Am I clear? Here it was 48, but in venous blood it is 52. It means during the transportations of carbon dioxide from arteries to the veins, tissue releases is around 4 ml blood. That is 4 ml carbon dioxide, right? Now see, all these events is carried out in the pulmonary capillaries. Now, once it reaches to the pulmonary vein, it ultimately reaches to the pulmonary circulation. So, if you see the characteristics of the pulmonary circulations or the reactions that is happens in the pulmonary capillary, it is just reverse of the reaction that is occurs into the systemic capillary. So here see, up to it's clear. Now see, suppose this is now pulmonary capillary. Here, according to law of diffusion, right? But before knowing that one, we must know about the reverse reactions that is occurs into the pulmonary capillary. What we have seen in the systemic capillary, HSB was formed where hemoglobin binds with the H plus ion. See, here in the pulmonary capillary, hemoglobin binds to the carbon dioxide. That occurs into the systemic capillary. As I already mentioned, the binding of both the oxygen and carbon dioxide depends on some conditions. Whether the hemoglobin and carbon dioxide presence in the systemic capillary or pulmonary capillary. In systemic capillary, as I have mentioned, in systemic capillary, the affinity between carbon dioxide and hemoglobin is much more than the affinity between oxygen and hemoglobin, right? But here in the pulmonary capillary, due to the higher affinity between oxygen and hemoglobin, it replaces the carbon dioxide from hemoglobin, right? Means here this HbCO2 is breakdown into Hb plus CO2. Now along with this Hb, now oxygen binds and it forms HbO2. So see, I repeat, in the pulmonary capillary, the affinity of oxygen towards hemoglobin is much more than the affinity between hemoglobin and carbon dioxide. That is why here, after the inhalation of gases, due to the more oxygen concentrations, it replaces the carbon dioxide from hemoglobin and binds with the oxygen. It forms HbO2, right? Now this HbO2 along with in pulmonary circulations through the exchanger, what we have seen, the chloride ion enters in systemic capillary, what we have seen, through this exchanger chloride ion enters and bicarbonate ion released out. But here again, as it is the reverse process, here it is the chloride ion again comes out and bicarbonate ion comes in, right? Now see, due to the influx of HbCO3 and due to the availability of oxygen, the pulmonary capillary shows different rate of reaction. So what is the rate of reaction here? HbCO3 again see, in presence of carbonic anhydrase, this carbonic acid again break down into some water molecules and free carbon dioxide, right? Now, due to the higher concentration of carbon dioxide here, and due to the binding of more and more amount of oxygen with hemoglobin here, there is a less amount of H+ ion remains in the inside of the cells, and the number of H+ ion here is less, and HbCO2 also enters into the cells, right? Both the HbCO3 and H+ ion again helps to maintain the pH of pulmonary capillary, right? Now, by the process of diffusion, this carbon dioxide returns back to the lung alveoli, and ultimately during expirations, this carbon dioxide released by normal expiration process, as well as if you see the rate of release of carbon dioxide, the amount of release of carbon dioxide is very high during forceful expiration. So here this is what is the exact mechanisms of carbon dioxide transport per bicarbonate ion, or as a bicarbonate, carbon dioxide transported out from the cells. Other than this mode of transport, what we have seen, around 5% is transported through the plasma. Due to the pressure gradient into the plasma, what we have seen, the alveolar pressure is little less than the carbon dioxide concentration present in the plasma. So only 5% of the carbon dioxide gas are transported through the plasma, and there is no need of, or there is no involvement of hemoglobin molecule. Only the pressure gradient is sufficient to remove the carbon dioxide or dissolved carbon dioxide. So this is what is the three different transport. The first one is as a bicarbonate form, and another one was, that is carbamino hemoglobin, carbon dioxide binds to the hemoglobin molecule, and third one only 5%, that is about the dissolved form of carbon dioxide transport. This transport of carbon dioxide are influenced by multiple factors. Along with these factors that we have already discussed, there are some other physical conditions or physical characteristics that affects the transport process. So what are the other physical factors? or physical conditions. Obviously, the rate of transport is affected by exercise, right? Rate of transport is affected by acidosis. Rate of transport is affected by alkali state. So it means some physical conditions along with some biochemical characteristics that combinedly affects the overall transport process. Not only the transport through the bicarbonate form, as well as they also affect the transport as the carbamino form as well as the dissolved form. This is just a very basic understanding how the carbon dioxide molecules gets transported.